kasih Pak ya.
The scientific talk is about to start. Please turn on the video on your webinar Zoom and be reminded to use the virtual background that the committee has shared. Mas Arga, izin memulai acara ya. Okay, ladies and gentlemen, allow me to extend our warmest greeting to the eminent speakers and guests today. Head of Research Center for Vaccine and Drug, National Research and Innovation Agency, Republic of Indonesia, Mr. Masteria Yunofil Saputra, PhD, Associate Professor at Griffith Institute for Drug Discovery, Australia, Mr. Rohan Davis, PhD, and Mrs. Yun Xiang Feng, PhD, Senior Research at Research Center for Vaccine and Drug, Mr. Asep Bayu, PhD, and Mrs. Penny Ahmadi. All the jet, ladies and gentlemen, a very good morning to you all. Welcome to the scientific talk. This is a monthly webinar series that organized by Research Center for Vaccine and Drugs, National Research and Innovation Agency, Republic of Indonesia. Today, our research group, Marine Natural Product, will host a webinar titled Current Trend in Marine Drugs Development. This topic will be presented in two seminars, one today and the other on September 5th, 2022. And at this moment, I am Asri Sofianti, Master Ceremony for this event today. On behalf of the committee, we extend our appreciation for you present today. As a start of our seminar, we will hear the national anthem of the Republic of Indonesia and request you all to take your seats. Distinguished delegates, ladies and gentlemen, now we move on to the speech segment, and that is going to be started with the opening remarks. Please be invited, Head of Research Center for Vaccine and Drug BRIN, Mr. Masteria Yunofil Saputra, PhD.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi Bapak Ibu semuanya. Thank you very much Bu Asri. First of all, I would like to say very thank you to Professor Rohan Davis and Professor Yun Fai that accept our invitation for the scientific talks for today about the marine drugs development. So, National Research and Innovation Agency, BRIN, we call it BRIN, is a scientific regulator in Indonesia through Research Center for Vaksin and Drugs BRIN has a strategic plans on marine natural product research from Indonesian seawaters. So 2 of my programs are directed to discover of active compound for antimicrobial resistance and Parkinson disease. Meanwhile, Griffith Institute for Drug Discovery has several key discovery areas, including drug resistance and also the Parkinson disease, which has also established the Nature Banks, a biodiscovery platform based on natural product extract and fraction derived from Australian natural resources. So based on the Griffith's experience, so we can get all of the information how we can maintain the biodiversity in Indonesia to get the marine drugs, marine natural products for drug development in Indonesia. And I hope with this scientific talk, we can make the collaboration in around Indonesia, not only in BRIN and Griffiths, but also from other university in Indonesia to collaborate to Indonesians, Australian archipelago marine natural products. I think that's the very important area for us to develop the marine natural products for drugs development. So thank you again for Professor Rohan Davis and Professor Yun Feng, and I hope all of the audience interesting for this, all of the topic, and enjoy the webinar. Thank you, Mbak Asri, Bu Asri.
Yeah, thank you, Mr. Masterio, for the opening speech. Okay, ladies and gentlemen, without further ado, let us begin our primary program today. We have a sharing sessions and have 3 topics to discuss right now. And at this moment, Mrs. Pehni Ahmadi, PhD, will moderate the seminar. Please allow me to provide you with her resume before we begin. Maybe the committee can help us to share the CV first. Okay, Mrs. Penny Ahmadi, PhD. She got bachelor degree in chemistry, Lampung University, and master's degree in chemistry, biology, and marine science from the University of the Ryukyus. She also holds a PhD in marine and environmental sciences from the same university, University of the Ryukyus. And now she is working as a senior researcher at Research Center for Vaccine and Drug. And here are some of her latest research experience. First, protein synthesis, in vivo imaging and cancer therapy, and then potent drug lead from Indonesian marine invertebrate to suppress breast cancer, and potential of latrunculin A, the bioactive compound of Latrunculia marine spawns from a relatively safe cancer therapy. Sorry. Here are a few of her publications. Synthesized pro-drug design enables biocatalytic activations in living mice to elicit tumor growth suppressions and antiviral activities of alga-based sulfate polysaccharide. Please join me in welcoming Mrs. Penny. Time is yours.
Thank you very much, Mbak Sri, for the kind introductions. And well, ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. First of all, thank you very much for Dr. Masteria for the opening remark as the head of the Research Center for Vaccine and Drugs, and the invited speakers and all the participants. And thank you very much for joining this scientific talk with the title Current Trend in Marine Drugs Development. Actually, for today's talk, we have 3 wonderful speakers, and we are very lucky to have them here among us. And this scientific talk will be done in a 30 minutes of talk for each speakers. And then after that, in the last session, we will have 30 minutes for discussion. During talk, you can write for all participants, you can write all your question or comment in the comment box, or later you can use a raise hand button in the discussion session to talk directly to the speakers. And right now, I would like to introduce to you for the first speaker. May I share my screen? Okay. So the first speaker is Dr. Asep Bayu. So he is right now a senior researcher in the Research Center for Vaccine and Drug in BRIN. And actually he got his bachelor degree in State University of Jakarta, and then he continued his master in a Master of Engineering in Department of Chemical Engineering in Bandung Institute of Technology, and then he got his PhD in Doctor of Engineering, Graduate School of Science and Technology, Hirosaki University, Japan. And his professional experience from 2015 to 2017, he is a research assistant in Hirosaki University, Japan. And then after that, in October to November 2019, he is become a visiting researcher in Farmamar in Spain. And then from 2008 to 2019, he is a junior researcher in Research Center for Oceanography, LIPI. And then 2020 to 2021, he is young researcher, Research Center for Biology, LIPI. And then right now he is a senior researcher in Research Center for Vaccine and Drugs, BRIN. And for further information, you can go and check for his website as written. And also the publication, you can also check his publication in his website. And right now, without further ado, I would like to invite Dr. Bayu to introduce his research theme. Or right now, he wants to introduce his research about Indo-Australian Archipelago, a biodiscovery hotspot of novel marine natural product. Dr. Bayu, floor is yours.
Okay, thank you very much for the introduction, Dr. Penny. I try to share my screen first. Okay, thank you for the time, Dr. Penny, and good morning, ladies and gentlemen who joined this seminar in Indonesia, and also good afternoon in Australian time to Professor Rohan Davis and also Professor Yun Feng. First of all, I'd like to say thank you to the committee for inviting me to be a presenter in today's seminar. And in this seminar, I will talk about Indo-Australian Archipelago, in which I will talk in the side of drug discovery, which is in these regions, mostly people interest to study about the biodiversity and also ecology. But in my view, that these regions is also rich for us to discover and to produce novel marine natural product. So my topic in this presentation is Indo-Australian Archipelago, a biodiscovery hotspot of novel marine natural product. Okay, this is a recent report published in Nature, which presents about biodiversity in the period of time. As we can see in here that since long time ago, the location near of equatorial line is rich of biodiversity. And if we move far away from the equatorial line, the biodiversity in this region will be decreased. And this is another information of how about the typical distribution of marine biodiversity in our planet. And as we can see here, that the location of our region Indonesia and some part of Australia is a hotspot for marine biodiversity. And actually, this is related with some parameter of environmental conditions in this area, which is good to support organisms, marine organisms, to grow, to stay, and to live in this area. For example, in here, if we look the global temperature distribution, the location in the equatorial line, especially in the Indonesia and Australia, in some part of Australia, has a warm temperature, and also this location is passed by the ocean flow, which contain high nutrition, like recent in here in the chlorophyll distributions. There are so many hotspots in this region. So this is the main reason why the region like Indonesia and some part of Australia is rich for marine biodiversity. Because of that, many researchers have interest to explore and investigate deeply about Indo-Australian archipelago. One of famous scientists that we know, Alfred Russel Wallace, he did expeditions and discovered the unique characteristic of flora fauna in Indo-Australian archipelago. He observed that in long time ago, actually, the island like Sumatra, Java, and also Borneo is in the part of Sunda land, and he observed that the typical characteristic of flora and fauna in this region is different with the typical flora and fauna in Papua region. And in the Wallace region in here, he observed typically the flora and fauna that live in here is endemic, that cannot be found in the Sunda or Sahur region. So the uniqueness of this typical characteristic has attracted many scientists, especially for marine biologists and also marine ecologists, and recently marine biographies to study about the characteristic of this area. And also not only terrestrial biodiversity for flora and fauna, Indo-Australian archipelago, because it is located in the equatorial line and passed by ocean flow with high nutrition. It also a hotspot for what coral live. There is a coral triangle area in this region, and also in Australia. Maybe later Professor Rohan Davis and Professor Yun Feng will present about many biodiversity that can be found in the Australia, and especially in the location called a Great Barrier in here. Okay, this is one of report from our colleague in the Research Center for Oceanography. They observe about the community, the marine organism community in part of the Indo-Australian archipelago area in here, and they observe that actually some communities have a typical region for their life in each area. So what is the relationship with the natural product in here? Usually when we see or when we talk about biodiversity, especially in marine. Most of people try to see how about the biodiversity, how about the ecology of the marine environment. But if we look more deeply about the marine organisms, we know that marine organisms also synthesize metabolites, either primary metabolites like carbohydrate, protein, or lipid, or secondary metabolites that are used for them to support their growth, to live and stay in their region. And for the marine, we know that marine has some characteristic like it has a strong UV exposure and it has wide temperature differences. For example, if we stay or we look the region in the tropical areas compared with the subtropical areas, the temperature totally different, and the coastal and also in the deep sea, the temperature will have a difference. And because of that characteristics, usually for marine organisms, they try to synthesize metabolites, especially secondary metabolites that are used for them to support their growth. And of course, in here, because some of marine organisms, like for example, sponges or porifera, which they live sessile or live in cannot move to other area, so they are depend on their biochemicals to growth and to live and to hinder the predator to hinder the predator attacking them. So all of these situations make them to synthesize the biochemicals that usually have a unique structure and also a unique characteristic that many reports have shown that the chemicals or natural products isolated from marine organisms have many multiple bioactivity that can be used by us by human to develop some economical products, like especially in pharmacy industry. For example, in here that one report published by Professor Luz, they found that in terms for us to discover compound with anti-tumor activity, natural product from marine organism has higher potency than natural product isolated from the terrestrial bioresources. So from these characteristics, we can see that the oceans with high biodiversity, that means it also show high potency for us to obtain multiple biochemicals that are useful, especially for us to discover drugs for pharmaceutical industry. Okay, this is, for example, one of report that show about the distribution of novel marine natural product from invertebrates. As you can see here, that previously I showed you about the location that become a hotspot for marine biodiversity. And if we look in here, yeah, the Indo archipelago region in here, which previously I saw you, it has a high marine biodiversity. It also this region we can get many novel marine natural products. And if we look the trends from 1990 to 2019, you can see the trend still increase. And I think that this research still have many potential for us to discover another novel marine natural product from marine. And this is also another report which presented about distribution of novel marine natural product from microalgae. Again, we can see from here that the Indo-Australian archipelago region produce many potential marine natural product from this marine organism. And the trend also similar. The trend for us to get the novel marine natural product from microalgae collected from these regions still increase. And this is a report when they try to see whether there is a correlation between marine natural product isolated from marine organisms and the typical biogeography in the area of Australia. And as you can see here, that even though in the same taxonomy, we can see the pattern of cytotoxic activity samples collected from that organisms are different depending on the location of the organisms obtained from this area. And for the antimicrobial active samples, also similar. It depends on the locations, which for which we sure that it has different environmental conditions will affect the distribution antimicrobial active samples that obtained from that organisms. And this is for the Indonesia also, we can see the trend for getting the marine natural product isolated from Indonesian marine organism is increased, which show the good potential of Indonesian seawater to get novel marine natural product. And this is some part of area in Indonesia, and mostly, as you can see here, that the eastern of Indonesia is a good location for us to obtain a novel marine natural product. Actually, this is correlate with the previous information that I shared, mostly because in these regions the biodiversity of marine organism is good. So the potential for us to get the novel marine natural product from this region is very, very high. And this is one of report that we published in our group, and we saw, as you can see here, that similar like before, when we investigate the marine natural product distribution in some marine organisms like soft coral sponges and tunicates, yes, similar like previous slide. In the eastern of Indonesia, the probability for us to get the novel marine natural product is high. Okay, this is some chemical natural products that have been isolated from marine organisms collected in the Indonesian seawaters. And in these natural products, they are named related with the locations of the sample collected in the area. For example, this is Manado peroxide, which is reported to have anti-protozoal activity with the IC50 around 3 nanogram per milliliter. This is collected from sponges collected in the Manado. So the active compound, this active compound was named as a Manado peroxide. And another one, for example, this is bitongolite, because this compound isolated from marine organism collected in the Bitung, which is one of area in the North Sulawesi. Lembain also, because this is collected from Lembe Strait in the North Sulawesi. And this is bicatite, which has a IC50, again NBT2 cell around 8.3, was isolated from biap. So this information show us about actually the Indonesian seawater has a potential for us to discover marine natural product with not only has novel structure but also has a novel bioactivity in the future. So in order to accelerate the research and development on drug discovery, National Research and Innovation Agency, or BRIN, BRIN, in here we established the Research Center for Vaccine and Drugs, and in this research center we focus to develop and to do the research about a vaccine and drug. And for the drug research group, we have 5 groups that focus to explore and discover the potential of natural resources, either terrestrial or marine resources from Indonesia, to find the active compound for drugs. And this is actually the main program in research center for vaccine and drugs BRIN. And we do some activities like expedition for collecting the micro and macroorganisms, and also we do chemical parts of this job, like bioactive screening and structural elucidations, and study about the mechanism action, and until we discover the active compound that we can try to formulate to obtain for drug formulations. And this is some research groups that part of drug development in the research center. And actually we BRIN previously in the research center for oceanography, or in the Indonesian Institute of Science, or LIPI, we have already established a natural product database from Indonesia. We call it Indonesian Marine Natural Product Database. And right now we try to continue to develop this marine natural product database, and we hope that the collaboration not only in BRIN but also with the other institutions from other countries to do the collaborations in order to find the natural products, especially from Indonesian seawater. So in the end of this presentation, I will give a summary. Indonesian archipelago is a hotspot of marine biodiversity, and this is comprising of large area with many potential, not only in terms of biodiversity, but also in terms for us to discover natural products, especially from marine. And of course, we cannot do it by ourselves. Indonesia cannot do it by ourselves, especially BRIN. So due to the large area of this one and the huge potential of this area for us to obtain natural product from, especially from marine. So we are open for collaborations in order to explore and to discover the natural product, especially from marine organisms in this area. And of course, so we hope that with later maybe Prof. Rohan Davis and also Prof. Kim Feng will present about their experience about the potential of natural product research in Australia. So probably in the future we can do some collaborations to do drug discovery and development research in terms of particularly marine and natural product from Indo-Australian archipelago. Maybe that's all from me. And if you have interesting or you want to discuss in detail, you can contact me by email, or you can also follow my work and my team in Marine Natural Product Research Group in our website. So maybe that's all from me, Dr. Penny, and thank you very much for your attention.
Thank you very much, Dr. Bayu, for the excellent talk. So if I could summarize your talk, is that the Indo-Australia region is the hotspot for marine biodiversity, and the marine natural product ratio to terrestrial natural product as a bioactive compound producer is 10:1 ratio. And the recent update also, you mentioned that Indo-Archipelago is high in marine biodiversity, and also there are reports on novel marine natural product from 1990 to 2019, and up to date trend is still increase in a novel marine natural product. And also you mentioned about macroalgae in Indo-Australia Pacific, and the trend is also increased. Thank you very much for the excellent talk. And right now, without further ado, I would like to introduce to you the second speakers. I would like to share his CV. So the second speaker is Associate Professor Rohan Davis, PhD, from the Griffith Institute for Drug Discovery. And his educational background is he got his bachelor degree in chemistry and biochemistry, University of Melbourne, and also he got his PhD in acid and chemistry. And his work experience from 1994 to 1996, research assistant on the AstraZeneca Griffith University Natural Product Drug Discovery Program, and then 2000 to 2003 as a postdoctoral researcher at the University of Utah, working on an anti-cancer drug discovery project. And 2003 to present, he is an associate professor at the Griffith Institute for Drug Discovery, and he's currently as a part of an Aussie Kiwi research team that publishes the well-known and highly cited annual Marine Natural Product Review in Natural Product Reports, and also manager of Nature Bank, which is a unique Australian biodiversity resource located at the Griffith. And his research area involves all aspects of biodiscovery, including the design and synthesis of unique screening libraries based on natural product scaffold. And without further ado, I would like to invite Professor Davis. Hi, Professor Davis, how are you?
Yeah, very well, thank you. Good morning.
Yeah, and today we would like to hear your talk about, wait a minute.
Could I just can't share the screen at the moment. Can you
sure.
give me rights to that, please? Thank you.
Please.
All right. Can you see something there?
Yes.
I just got to move this. Can you see the full screen now?
Yes, right now you can see that.
Wonderful. And you can hear me okay, obviously?
Yes, very clear.
All right, wonderful. Well, I'll
Yeah.
get underway. And firstly, thank you to BRIN for the invitation to present today.
It's our pleasure.
Lovely to be talking to lots of different scientists around the world, and there's lots of great scientists in Indonesia. So happy to be sharing some of my experience and some of the things that my research group have been doing over the last 10 plus years. And thank you for your kind introduction as well. That was great. So I'll get underway. I believe I've got 30 minutes and it's 12:48 now. So hopefully I won't take all 30 minutes. So my topic today is Nature Bank Adventures in Marine Natural Products Drug Discovery. And in the next 25 minutes, I'll give a bit of an overview of our institute, Griffith Institute for Drug Discovery. Ewan, I hope I don't steal too much of your thunder there. I'll skim over the grid stuff and then get to my stuff so as not to impact your talk too much, Ewan. So can you see the second slide okay?
Yes.
All right, great. So this is the place that Ewan and I spend a lot of our time at. It's the Griffith Institute for Drug Discovery, located in Brisbane, about 5 kilometres south of the CBD. We've actually got 2 buildings at GRID, and they both house approximately 110 researchers, as well as admin staff. We've got over 60 HDR or PhD masters and honours students, and the rest of the people that work at GRID are typically chief investigators like myself or research leaders, and we have our own specific research group. Mine, I've got 5 PhD students and a couple of undergraduate projects running at the moment. So there's a number of research themes running at GRID. Both, of course, natural product drug discovery is the history of this institute, but we're doing different things now, as well as still natural product drug discovery. We've spent a lot of time looking at terrestrial organisms such as plants and mushrooms, but we also spend a lot of time on the marine environment, and that's, I think, why I got the invitation to this talk today. So cancer, infectious diseases, neurological conditions. Yun's going to talk more about neurological conditions later today, but for the past 10 years, my group's done a bit of everything here, and I will focus on 1 or 2 specific projects. 2 of the unique facilities that we have at GRID is Nature Bank, which I'm the manager of, and I'm going to tell you about that over the next few slides, and Compounds Australia, which is a national compound repository that maybe I think Yun will be talking to you a lot more about in her talk. So this is a very busy slide and it's got a lot of text on it. I'm not expecting you to read it as I chat, but suffice to say that this is a brochure, a Nature Bank brochure that we send to potential collaborators. It explains some of the metrics around Nature Bank. It talks about our aims and what we apply our resource to. As you can see there, it's not just about new drugs anymore. We look at agrochemicals and even nutraceuticals. The next page has a little bit about how we work with industry, our services, and how we do research collaborations. So if anyone that's listening today types in the word Nature Bank and Griffith University or GRID, you'll be taken to the homepage of Nature Bank, where you'll see some videos that I've presented. I present. It shows a list of our recent publications, and you can download this flyer and read it at your leisure. So, Australia, like Indonesia, is considered a mega diverse country on this planet. And the crazy thing is, with these 17 mega diverse countries on Earth that are highlighted green on this map, they cover less than 10% of the Earth's surface, yet contain greater than 70% of the Earth's biodiversity. And so, as ASAP was talking about, both Indonesia and Australia are very lucky because of the biodiversity that we have in our countries and our coastlines, of course, our waters. And typically biodiversity equates to chemical diversity. So if you're doing drug discovery or any type of biodiscovery, you need the biodiversity because you need the chemical diversity for the high throughput screening campaigns that are typically run. So Nature Bank in a nutshell, we went in the 1994, the founding director of our institute, Professor Ron Quinn, set up a collaboration with the pharmaceutical company AstraZeneca. This was a 14-year collaboration where we worked exclusively for AstraZeneca, looking for new natural product leads or drugs from a variety of different human diseases. It included infection, cancer, cardiovascular disease, gastrointestinal, and pain as well. And so before we could start any natural product drug discovery, we had to go and collect a lot of samples. And we focused on the state of Queensland because this is one of the most biodiverse states in the country of Australia. Western Australia, which is, as the name suggests, on the western side of our great continent, is also a very highly biodiverse state. But in Queensland, we have really unique ecosystems. As Sepp mentioned, we've got the Great Barrier Reef, which is a real treasure and an icon for us. And we've collected a lot of samples there. Most of our marine samples come from those waters. But we also went and collected in the terrestrial ecosystems, whether they be subtropical or tropical or even desert environments. And some of my recent projects have been on desert plants that have been under investigated to date, for example. So we have 30,000 biota samples. The breakdown is about 2 to 1. For every 2 terrestrial samples, we have 1 marine. So that means about 20,000 samples of plants and mushrooms, and the remaining are marine invertebrates, such as corals, ascidians, tunicates, algae, etc. So we've taken a portion of those 30,000 samples and made extracts, lead-like enhanced extracts. And this work, how we actually go about making the extracts and the fractions that I'm going to tell you about in a moment, this work's been published in Journal of Natural Products in 2012, if you want to download that paper. It talks about the specifics about how we went about creating the extracts and how we also limited the amount of lipophilic material that was in the extracts. So each of these 21,000 extracts, a portion of these in DMSO has been injected onto an analytical HPLC, reverse phase, C18, and then separated into a number of fractions. And back in the day, it was 11 fractions that we collected and made our fraction library. But in more recent times, we've only collected 5 fractions. So these are timed fractions. Each of these fractions is dried down, resuspended in dimethyl sulfoxide, and then added to microtiter plates or Abgene tubes, as you can see there. Everything is barcoded so that we can track every sample from the moment we collect it to the moment that that extract or fraction goes on a microtiter plate to the moment it gets sent to a company or a collaborator overseas or within Australia. All the samples were GPS located as well. That knowledge or that data is held by the Queensland Museum and the Queensland Herbarium, who I need to give a special shout out to, because the museum and the herbarium were the ones that for many years were out in the bush or on the reef collecting these samples. And they're the taxonomy experts that actually identified these samples for us. So Nature Bank works in a couple of different areas, but I'm going to tell you 2 stories today, 2 on human health and 1 on animal health. So this is all published data that I'm going to share with you. So hence I'm happy to have this broadcast and happy to answer any questions about it. One other comment that I would make about Nature Bank is that we do it in an ethical and sustainable manner. We're committed to the Nagoya Protocol that was signed many years ago. And we have benefit sharing agreements in place with the Queensland Government, the Queensland Herbarium, and the Queensland Museum. So that if we discover something, it's not me that benefits from that. It will be Griffith University, but it will also be the Queensland Government. It will be the Queensland Museum and Herbarium. Everything gets shared, although the sharing funds might be very small in amounts. It doesn't matter. We're trying to do good collaboration here, and benefit sharing agreements are critical for that particular aspect of biodiscovery. So our vision, we're actually starting to live our vision, because Nature Banks, I took over as manager in 2017, and I regenerated the libraries, made fresh materials, both the extracts and the fractions, and then started to approach companies, but also academics within Australia and overseas, about accessing this unique resource. And so we've sent screening plates, whether it be just a couple of thousand samples or 105,000 fractions. We've sent those overseas to a variety of different people. And at the end goal, though, our vision is to produce a product, something that's commercial, whether it be in the nutraceutical, agrochemical, or biomedical fields. We haven't done that yet, but I believe we've got everything lined up in order to give us the best chances of success in this particular area of research, which is firstly very competitive, but also very high risk. So this is the biodiscovery workflow. I hope you can see the top of the slide here. But our biodata samples have been made into, as I mentioned, the extract and fraction libraries. Typically, high throughput screening is undertaken. And then once we identify the hit fractions and extracts, we do bioassay guided fractionation, a very old way of doing it, but still a very good way of identifying new bioactive compounds. We do mass-directed isolation, NMR and UV guided. We do all of that, and at the end of the day, we end up with lead compounds or hit compounds. Those lead and hit compounds are what the companies, as well as the academic collaborators, are after. We have expertise, of course, in purification, but also structure elucidation. So the interpretation of 1D, 2D NMR data, for example, is something that my group's particularly skilled in. And we come up with, we elucidate structures such as those shown here at thioplascortone A. So this is quite an old story. But it's a great story, and it continues to evolve. This molecule was purified from a Great Barrier Reef sponge, Placourtis liter. This was work that I went back when I was in the lab. I'm not in the lab anymore. I'm spending a lot of time coordinating Nature Bank biodiscovery efforts and writing papers and grants. But at the time I was in the lab, and we purified this compound and worked out its structure. This molecule had a novel skeleton. So the ways the atoms are all connected was very unique. And we got this published in Journal of Organic Chemistry back in 2013. It was a result of Medicines for Malaria venture funding that we received to screen our fraction library against Plasmodium falciparum, both drug-resistant and drug-sensitive strains. And you can see that this compound, in vitro anyway, had quite outstanding potency, nanomolar activity against both the DD2 and 3D7 strains, some toxicity against the human embryonic kidney cell line, but there's a good selectivity index there that meant that I had to go and write a grant and get some money to do some more research on this particular molecule. So we went and this is work that was collaborative with Ron Quinn, and Rebecca Power was the clever synthetic chemist that came up with this scheme. There was a group of synthetic chemists working on this. She was the first author. So it's an 8-step synthesis for thioplascortone A and a 7-step synthesis for thioplascortone B, which I haven't shown you, but it's the unsaturated thiazine analog of thioplascortone A. So with these molecules in hand, we could then progress. We've started mechanism of action studies, but we could also progress into animal studies. And this is one molecule, RAD214. It's not the natural product, but it was one of the MedChem optimized analogs that we identified. And it was very stable in mice microsomes, nicely soluble still as the acetylated version of thioplasmine A. And as you can see there from this curve, this is a Peters 4-day test, which is testing actually against Plasmodium berghei, which is, this is a strain that infects rats and mice. And you can see there that RAD214 actually suppressed parasitemia when it was injected. Now, the problem is, though, you can also see, hopefully, the concentrations that RAD214 was injected into these animals at. So 32 mg per kg is quite high, and comparing it to the gold standard that we had, which was chloroquine at 1 mg per kg, we just didn't have something that was active enough at that point in time. We've published that work subsequently. This is back in 2015 in Organic and Biomolecular Chemistry. There's other work now going on with the thioplasmine core. We're actually doing what's called late-stage functionalization on this molecule, and you're going to see some papers come out from my group in the near future about that. We're trying to further optimize the molecule. I should make the comment that thioplasmine was trialed for oral bioavailability, and that's probably what stopped the project at that point in time, because even though we had injectable activity in the animals, oral bioavailability was lacking. And that's what's needed for new antimalarial drugs. You need to be able to pop a pill rather than have an injection to cure your malaria infection. In the interest of time, I'll move on, and I want to talk a little bit about something that we also do at GRID, which is we take marine natural products as well as plants, and we do semi-synthetic studies. So this is work that my PhD student, Rohitesh Kumar, performed a number of years back. We isolated synaptolin F, shown at the top. It's a beautiful molecule with exquisite three-dimensional shape with this spiro acetal unit. We isolated almost 0.5 gram of this molecule from a sponge, Hyatella species from the Great Barrier Reef. And then Rohitesh went and made some analogs. And R1 and R2 that you see on that slide shows you some of the points where he varied the chemical structure. And then doing classic medchem, those analogs that we made, we went and screened them, and we were interested in its ability to inhibit prostate cancer for this particular paper. Found some interesting attributes which are shown. We've got some great images of what these molecules are doing inside the cell, and some hints into the mechanism of action of these molecules. So this is the paper. If you want to download it, it came out in 2020, and it's research that's done with Professor Colleen Nelson's group at the Translational Research Institute here in Brisbane, and Colleen leads up a team at the Princess Alexandra Hospital at TRI. All right, so we've got the Davis group have been isolating pure compounds for almost 2 decades now. Every time we isolate a compound, we put it into the Davis Open Access repository, which is stored at Compounds Australia. And this library over the last 20 years has been sent out to researchers in various parts of the world. Most of these compounds have been published. You've got some really unique chemotypes, many of them from the marine environment. So that library, as well as the Nature Bank extract and fraction libraries, there's over 11 different collaborators that we're working with at the moment, 9 universities and 2 companies. And these are some of the areas in the biomedical realm that we're working with. Okay, so I wanted to shift gears a little bit and talk about an agrochemical project that we're running. This is work that we're doing with our collaborators at Melbourne University. They're interested in finding new molecules from nature that will inhibit the growth of the nematode Haemonchus contortus. This is a parasite that is present in both temperate and tropical climates, and it really causes some problems in in ruminants and livestock, such as sheep and goats. It can produce death in young animals, and it just lowers, it has a major economic impact. In Australia, it's believed to have an impact equating to a roughly $436 million a year. So devastating disease for livestock. So that's no good. Can we find new drugs from the marine or terrestrial environment that might stop this parasite? A very complicated life cycle. I don't think I've got time to go into that now. I think I've got 5 minutes left for my talk. Anthelmintic drug resistance is present, unfortunately. You can see some of the synthetic drugs as well as some of the natural products, ivermectin and moxidectin. These are natural product derivatives that have been very successful for the treatment of Barber's pole worm, as Haemonchus contortus is also known. But since the year of the drug being approved for treatment in livestock, there's always been resistance developed, 4, 9, 7, 2, and 7 years after the release of the drug. So ongoing resistance issues are a problem, and that's why we need to find new drugs. Very quickly, this is the start of our journey for new anthelmintics from Australian biota. There's some plants, a plant paper shown there. Robin Gasser is the lead professor down at Melbourne University that we publish with. And then the example on the right is some known marine natural products, phromiomycalin and halaminol A, which were isolated from marine sponges from Australia and shown to have decent activity against Haemonchus contortus. Subsequently, from a preliminary pilot study that we did with Gasser, he went and screened 7,616 marine extracts in the Nature Bank extract library and found 58 extracts, and one of them I want to talk about very briefly, and that's what I'm going to finish on today. Citronia is a really unusual sponge genus. We've only got, I think, 6 in our collection, and some fractions from this extract showed really good anthelmintic activity. The most active fraction, which was fraction 50, we ran UHPLC-MS to work out how complex these mixtures were. We also, of course, did a lot of proton NMR spectroscopy. And there were 2 molecules that we identified following dereplication steps that involved accessing Marine Lit database that we have a subscription to. This is the beautiful proton NMR that of dicidothiazole that my grad dip student ran and identified the molecule, confirmed this molecule shape. Sorry, not just the shape, the planar structure, as well as the relative stereochemistry and the absolute stereochemistry. The relative was determined by 1D and 2D, but like I said, it had been previously identified. We were just confirming the structure was the earlier reported marine natural product molecule. And we're also fortunate enough to get a crystal structure. So there's the crystal structure. Decent activity for dicidenin is the most active, dicidothiazole less active, but it produced a really interesting phenotype against the parasite, the Barber's pole worm parasite. Here's where some of the libraries have been sent. And then finally, I'd just like to talk a little bit about impact. Impact's been, academics around the world have been pondering the word impact and how to measure it for a long time. It's becoming the new buzzword again. Whether you like it or not, you have to think about research and your impact to the world. And Nature Banks, we're having some nice success. We've also just recently identified a natural product from a sea squirt collected off the coast of Rockhampton, which is in Queensland here, that also has anti-cancer activity, and that's going into animal studies. It's started animal studies last year, and we're also undertaking ADME and DMPK studies. So this was an article that appeared in this website, Medical Research Discoveries. You can go to that website, and this is the Queensland Museum collecting this, a sponge for us from Nature Bank. That's the photo in the background. The other thing I wanted to give a plug for was this review that I'm part of. It takes me almost 6 weeks of my life every year to write it. I'm one of 5 authors. Tony Carroll is another marine natural products chemist at Griffith, who I work with on this paper. Tony's the corresponding author. And we've got these three Kiwis, Brent, Robert, and Michelle, who are wonderful to collaborate with. Last year, we put out this paper, and I think there was, what was it? Just trying to quickly read. I think it was 1,400 new marine natural products were reported in 2020. And these are some of the nice sites that equates to some big impact for this review for us. It's the review actually in Google Scholar has just clicked over the 600 sites. That's the first review that Tony and I wrote in 2018. So for those of you who are older than me or my age, you'll know that these reviews were started by John Faulkner at Scripps in the US. And then over the years, other authors have contributed. And this is the one review that I'm contributing to at the moment. So lots of people to thank. I don't have time to list them all out, but I'd like to thank everyone for logging in and listening to my talk. And I'd be very happy to answer any questions during the discussions that will proceed. Thank you.
Wonderful. Thank you very much, Professor Davis. If I would like to summarize your talk, that in grid, you're still working in marine natural product, but also in terrestrial, which is you collect from tropic, subtropic, and also desert, and also you work on mushroom. And you also use nature banks, which help chemists to discover novel compounds. And what else? You also isolate thioplocarton A in 2013. And then it's really interesting structure because it's new cyclotron. And then after that, 1 year later, you did a total synthesis on this thioplocarton A and also with the thioplocarton B. And then after that, you also did a mice kind of mice therapy in anti-malaria. It's anti-malaria, but somehow the bioavailability is lacking. So you try to do a semi-synthetic, another semi-synthetic of natural product. And then in addition to that, you also have some projects on agrochemical project with working on parasitic, and also you work on anthelmintic drug discovery. It's really wonderful research, and I hope it's going well for it, and we are looking for a further result from your lab. Thank you very much.
Thanks, Penny. Great summation. I'll just turn my microphone off. You've got sharing capabilities again. I think I stopped sharing.
Yes, you
Right. Thank you.
Yes. Okay, right now I would like to introduce the third speakers. Let me share. So she's Associate Professor Yoon Jung Feng, PhD, a research leader in Griffith Institute for Drug Discovery, or GRID. And her background, educational background is in PhD in marine natural product chemistry in James Cook University. And her work experiences from 2000 to 2003 as a postdoctoral research in anti-cancer natural product at the University of Canterbury. And from 2004 to 2013, she is a research fellow position at Griffith University, leading over 20 commercial in confident natural product drug discovery project. And from 2014 to present, she is an associate professor and currently led in the natural product and traditional medicine group at the Griffith Institute for Drug Discovery. And her research area is combine the massive chemical diversity of natural products with the power of high throughput, high content screening to expedite the search for biologically active molecules. And right now, I would like to invite Professor Feng.
Thank you.
Hello, how are you,
Hello.
Professor?
Hi, how are you? Hi, everyone.
Thank you. Hello. Right now, please, the floor is yours, Professor Feng.
Okay, I'll try. It says I cannot start screen sharing.
I'm sorry, so please share your screen.
I think you can see. Can you see? Please let me know.
Yes, we can see, but it's not the full one. But can you share with the other type of slide? I mean, the total full screen one.
I have shared full screen. Is it better now?
Yes, but it's not the full screen, but we can see some another notes there. So I mean, can you share the, could you please end slideshow first, and then let's start with the full screen one.
Okay, let me just
I'm sorry.
exit.
Yes, I can see in a slideshow.
Yep. Better
I think
now?
it's still the same.
Okay,
Sorry.
let me try one thing.
So, sure. Could you go to the slideshow tab?
Stop sharing, then try again.
Oh, sure.
Okay.
I'm sorry.
No, you're right. Let me see. Share. Still not good?
Yeah, it's not good. I mean, not full screen yet.
Ah, I see. I'm not sure what's the problem. I'll close it first, then open it again. Sorry about this.
No worries.
Okay. Better? No. Still not. What can you see? What can't you see?
It's your slide, but you also show our next slide and notes, something like that. It's not.
Oh, I see.
Yes. Sorry. So.
Yun, do you have a PDF of this presentation? I just wonder.
No, I have the animation. If I turn it into PDF, would be something will be covered. Would be quite
Right.
difficult. Yeah,
Okay.
if I go to the hide the presenter view, I can't see anything.
Yeah. Okay.
So I'm not sure
Yeah.
why.
Because about 2/3 of the screen has got the slides that you want to show, but it's just 1/3 of the screen has got next slide and and your notes. As Penny
Okay.
mentioned.
Let me see if I can increase this. So I've tried just to hide the presenter view, then I can't actually see my screen anymore.
Okay.
Yeah.
Maybe you might just have to run with that. Have you got many notes?
No, not really. No.
Okay.
Is that okay? I just go with this.
Okay. No worries.
Yeah, sorry about all this. Well, thank you very much for your kind introduction, and thank you for the opportunity to present some of my research here. I can see it's quite a large audience. Thank you so much for joining us. So today I'm going to give you a very quick introduction of one of the research schemes I'm running in my group, which is the identification of molecules to study Parkinson's disease using we call psychological profiling based phenotypic assay. And before I go into that, I want to give you a quick review on my research as a general sense. So I'm leading a research group at Griffith Institute for Drug Discovery, known as Natural Products and Traditional Medicine. So we're looking at bioactive natural products in many different disease areas. Parkinson's, of course, is one of the focus we're looking at. We're also working on anti-infectious disease and collaborating with another academic staff within Griffith University, looking at tyrosinase inhibitors in the application of treating hyperpigmentation disease, as well as in agriculture and cosmetic applications. So come to Parkinson's disease, I guess one of the grand challenges for Parkinson's disease is really the lack of understanding for the disease, and also more importantly is the lack of disease models to reproduce this complex biological signature of Parkinson's disease, as well as per understanding of what's going wrong when people develop Parkinson's disease. So as a result, in the last 5 or 6 years, we, as a group called Parkinson's Network within Griffiths, we have established a cell model, also known as a cytological profiling-based phenotypic assay, and really trying to understand the biology of Parkinson's, and also to trying to discover small molecules, use a probe to study the mode of action, as well as to a target identification, really with the goal to find molecules can be used as a drug to treat a disease. And so far, we have published a few papers, which are listed here, focusing around this platform we have developed. As you can see, we started around 2015 on this project, and it's still ongoing. I'm only going to report you some very initial results we have achieved so far. It's still, for us, a long way to go in terms of finding that initial molecules can be used as drug to treat a disease. So if you're interested in this model, please check it out. There are a few papers there I've listed on the slides. So in terms of this model, what we used is a patient-derived cells, also known as, also known as human olfactory neurosphere cells. The abbreviation is HON cells. And those cells are primary cells harvested from Parkinson's patients. They are from the olfactory epithelium. So we extract the cells from the patient, then we culture the cells, and we develop these cytological profiling methods. The goal is to look at whether we can use this cell model to study Parkinson's disease to find molecules which help us understanding the mechanism of action. So our research has shown these cells are very good model in terms of reflecting on Parkinson's disease pathology. We consider it as really a window and a door to the brain. We can use these cells because it's very difficult to access brain cells, but we can use these HON cells as a model to study the brain function. In this instance, is the Parkinson's disease. So at GRID, we have a neurobank known as neurobank. We have a collection of about 200 cell lines harvested from patients suffering different diseases. I have listed here, for example, motor neuron disease, Parkinson's disease, mitochondrial mutational disease. All these diseases are associated with brain function and also aging. So we can use the cell lines to establish assays to study the mechanism of this disease. So what is phenotypic assay? A phenotypic assay is really based on the cells harvested from patient. We can culture the cells, we can stain the cells using different fluorescence probes. We can also stain the cells with antibodies. Then we can visualize some cellular compartments, for example, mitochondria, tubulin, early endosome, lysosome, and cell morphology. So it's really give us the opportunity to see how these organelles, which are very much implicated in Parkinson's disease, and how we can use small molecules to manipulate these cellular organelles in order to study the function of the molecules as well as the underlying mechanism of Parkinson's. So in these assays, we can measure 38 parameters related to nucleus, cell morphology, alpha tubulin, mitochondria, and 3 other compartments of the cells. So we're targeting these cell organelles. The reason for that is the study have shown those organelles are very much important in the development of neurological disease, particularly Parkinson's disease. So here, I will quickly go through how we run the assay. We have the cell culture, which we robotically transfer the cells into 384 well plates. We will culture the cells at 37 degrees, 5% CO2 for 24 hours. Then we'll do the cell staining using fluorescent probes. And then we can acquire image using this automated imaging system known as Operator, which is a high content imaging system. It's a live camera, can take picture of cells and focusing on different cellular organelles and capture the changes in the cell functions as shown a few photos here. So once the images are captured, we can analyze these images, do a segmentation of the image, and then extract the features, focus on 38 parameters related to the functions of mitochondria, early endosome, lysosome, cell skeletons, and the morphology and autophagy. And once this is completed, we can analyze the data and transfer the data to also conduct a statistical analysis and generate a cytological profiling as shown here to show us when we treat the cells with small molecules, how the cellular organelles are changed, are they related to Parkinson's disease. So here I've shown you a phenotype of 2 molecules. As you can see, the photos on top really show you the cellular changes and the colors representing the staining with different fluorescent probes or antibodies. And we can quantify the cellular change by using our algorithm log-2 ratio, which is compared with the control group. We treat the cells with molecules, and we can collect the data and compare with the control group, which is DMSO treatment. And then we can transfer the data into the log-2 ratio and see how the molecules can perturbate the organelle functions. As you can see, yellow color showed perturbation is negatively changed, and blue color showed perturbation as positively charged. So then we can generate a heat map. This is cluster different parameters against the molecules and class those compounds with similar phenotypes together against the different organelles. So we can generate a heat map against many different compounds, against many different parameters, and see what kind of phenotype we can observe after we're treating the cells with small molecules. So we conducted a high content or high throughput screening campaign targeting Hong cells. We tested just over 4,000 fractions, and we have around 100 active fractions. And then we conducted the second repeat, biological repeat, and we only had 20 confirmed. So it's quite low. So after looking at the data, we considered as 9 fractions and 8 fractions. That means it has certain perturbation. Log-2 ratio has to about 1.5-fold change of treated group compared with the control group. So one of the hit fractions, 2 fractions I want to talk a little bit more is coming from marine sponge. With the taxonomic information here, Jasper's splendens. So here showed you HPLC traces of this sponge extract. This actually is the nature bank fractions Rowan discussed earlier. As you can see, the 2 fractions highlighted, which is fraction 2 and the fraction 10, showed quite a nice perturbation against organelles we are interested in. As a result, this sponge material or extract was selected for a large-scale isolation work. So given the phenotypic assay is really time-consuming and complex and generate large amount of data, the approach we take for this chemical isolation is NMR fingerprint-guided isolation. So this is widely used in my group. So we use proton NMR as a guide in isolation. As you can see, this is only an example to show you the concept. So the top trace show you the proton NMR of one fraction. Then by looking at the signals in the proton NMR, we are able to isolate 5 compounds. The 5 compounds together pretty much demonstrated all the major compounds from this fraction. So we're quite happy we managed to isolate pretty much all the compounds from this fraction. So this approach we know as proton NMR fingerprint guided isolation. So we have bioassay guided isolation, but in this instance, because of the complexity of the assays, we end up using the proton NMR fingerprint guided isolation. So as you can see in these slides, fraction 2 was quite an active fraction. We managed to identify the 4 major compounds as shown in NMR here. And the later fraction, which is fraction 10, we managed to identify 2 major compounds, but large-scale isolation enabled us to really identify the 22 molecules from this marine sponge. So as you can see, it's really weak presence of those compounds. However, we managed to identify quite a few nucleosides and analogs. We managed to isolate some indole alkyls and bis indole alkyls from the middle of the trace. We also isolated 5 peptides from later fractions. In total, we identified 22 compounds from this biota, and 3 of which are minor analogs of known compounds. So from other feed fractions, we also isolated 255 compounds. This is not from the sponge, from other heat fractions. And 84 of those 355 are major metabolites. As you can see, 20 from Jasper's splendens and 30 from a very well-known traditional Chinese medicine known as ginseng, and some other compounds from plant material. As you can see, the structures are quite diverse for this particular project. Then we run the phenotypic assay on the 84 major metabolites, which we have plenty material with, and we looked at their phenotypes and clustered those 84 compounds into 6 different bioclusters. That means each cluster have similar phenotypes against the organelles we're interested in studying Parkinson's disease. So I'm not going to go into details in terms of 84 compounds, but I do want to highlight a number of compounds which showed quite a strong perturbation against organelles we're interested in. So here showed you diverse structures have similar phenotypes against tubulin, mitochondria, the autophage and the lysosome. As you can see, even though they have quite diverse structures, but they showed very similar phenotypes, which indicate similar mechanism of action. So this major compound, which is jaspermycin from the sponge, also showed quite a nice perturbation against lysosome parameters, alpha tubulin, mitochondria, and autophagy, as you can see in these slides. And very pleasingly, we identified one compound, which is a known compound and known as bognolin, which is has been reported as anti-cancer agent. These compounds more or less reverse the effect of rotenone, which is a very well-known neurotoxins. As you can see from this bar chart, the y-axis is a log-2 ratio, and the x-axis are 22 major parameters related to different organelle functions. Particularly in mitochondria, we know rotenone is a mitochondrial complex 1 inhibitors, which can cause cell death, which is well-known neurotoxins. So this molecule more or less reverse the mitochondrial function against rotenone, which we are very excited about. So we have, by running phenotypic assay against 85 compounds, we have accumulated large volume of data. So we know which groups of compounds have what certain phenotypes. And now what we really want to know is to interpret those phenotypes. What are the underlying mechanisms, right? So against those 38 parameters. So what we just started early this year is to use NMR metabolomics to see what different phenotypes, what are the mode of action, and can we identify drug targets by looking at different phenotypes. So this is a very quick summary on what is involved in terms of target identification. That's drug target identification by NMR metabolomics. Essentially what we do is we culture the cells in the presence of the drug or without the drug. Then we can get the cellular, extracellular culture medium, then we can run NMR, proton NMR. Then the NMR we can input into the software to undergo multivariate data analysis. And this data analysis can further undergo a PCA analysis, which is the principal component analysis, and obvious discrimination analysis. We will be looking at the outliers and also potential biomarkers. But for us, more importantly, we want looking at metabolic pathway change, whether the molecules can induce certain metabolic pathways, and this will lead us potentially to discover targets for treating Parkinson's disease. Okay, we have some proof of concept data so far. And what we did is we treated cells with the rotenone, which is our positive control, which is also a very well-known mitochondrial complex 1 inhibitor. We know its mechanism. We want to see whether this NMR metabolic will lead us to the same molecular mechanism. So we culture the cells, Hon cells, with rotenone, and then we extract the extracellular fluid for NMR analysis. Here I show you a few NMR spectrum. As you can see, they are very, very complex. But by using certain software, we can identify all the molecules present in the cell culture. For example, quite a few amino acids, sugars, and small endogenous molecules. Then we undergo further analysis using obvious discrimination analysis, and we can predict the compounds. In this analysis, the positive peak indicate metabolites change, and the positive peak indicate the change metabolites more abundant in the treated group, and negative peaks indicate the metabolites more abundant in the control group. And the hot colors are metabolites associated with this cluster, and cold colors means no difference. By looking at this analysis, we can conclude a decreased level of alpha and B glucose when the cells are treated with rotenone, which is the known neurotoxin, and that indicate rotenone increased glucose consumption in Hon cells. And like I mentioned, rotenone is mitochondrial complex 1 inhibitor. It can block ATP production while suppression of oxidative phosphorylation. So our data so far is the decreased level of alpha and beta glucose indicate the involvement of rotenone in the glucose consumption in the Hon cells, which really in line with the literature, which means rotenone is a mitochondrial complex 1 inhibitors influence ATP mechanism. So we're quite pleased with the initial results. The next step we're going to do is to culture the cells with the selective small molecules with a different phenotype, particularly compound which reverse rotenone effects, then to see whether we can identify certain mechanism of action. In addition to phenotypic assay, we also developed a number of functional assays to study, to see whether the compounds can have clear indication on Hon cells. So one of the functional assay we are in the process of developing is the mitochondrial membrane potential assay. a PhD student just started in April. So his project is to really test the compounds for cytotoxicity, and also using this functional assay to test the compounds, whether they can be a modulator for mitochondrial function, because we truly believe mitochondria is a very important organelles involved in Parkinson's disease, and it's also widely involved in some metabolomic disease as well as cancer. And the secondary functional assay we have established is the alpha-synuclein aggregation assay. Alpha-synuclein is well known, involved in neurodegenerative disease. The aggregation of alpha-synuclein lead onto the pathology of neurological disease. So what we have established this assay, we are able to test the molecules to see whether they can stop the alpha-synuclein aggregation. So in terms of alpha-synuclein, we also have a technology in the institute, which is using high-resolution mass spectrometry to detect the native protein and to study the function of native protein with ligands. So using the technology we have here, we have established a alpha-synuclein binding study. So as I said, alpha-synuclein is really, the aggregation has a lot to do with the Lewy body, which is the hallmark for Parkinson's disease. So what we have established is to express and purify alpha-synuclein and then detect the alpha-synuclein by using high-resolution mass spectrometry. So as you can see, we can inject the native protein through the mass spec. We can observe the native protein in the multiple charge. In this instance, alpha-synuclein, the molecular weight is 14,500 Dalton. We can observe the 14 charge and 13 charge of alpha-synuclein. And the idea there is to incubate the alpha-synuclein, and we are able to observe the complex of alpha-synuclein with the small ligand. And then we can calculate the molecular weight of the small ligand by subtract the molecular weight and times the charge. So that's the idea. And so far we have tested a number of small molecules. We isolated from ginseng, which is a very well-known traditional Chinese medicine, which is well-known, good for brain function. We have identified a number of ginsenoside by using FGMS to observe the binding of the ginsenoside to alpha-synuclein. And here showed you about 10 structures we have identified which are the ginsenoside bind to alpha-synuclein. And these molecules will be tested in the alpha-synuclein aggregation assay to see whether the binding to alpha-synuclein lead onto its property or their properties to stop the aggregation of the alpha-synuclein. So that is really a very quick and summary on the Parkinson's research in my group. If you're interested, I'm more than happy to give a bit more details. This is really a very quick introduction to the audience and see what is happening in my group in terms of Parkinson's research. So before I conclude, I wish to draw your attention to Compound Australia facility within GRID. I'm currently the academic lead for Compounds Australia. One of my objectives to facilitate the collaboration between the facility and international or national collaborators. So to put it very briefly, Compound Australia is an Australian national facility, a compounds repository facility. Within the facility, we have about 1.5 million compounds. They are small molecules, right? Small molecules used for drug discovery purposes. In terms of open access collections, we have about 33,000 scaffold collection and over 8,000 drugs which are on the market that can be used for drug repurposing research. We also have open academic collections, that's more than 40,000 compounds that can be sent to scientists around the world for drug discovery purposes. And of course, Nature Bank extract and fractions are also stored at Compounds Australia. So if you're interested in finding out more, you can check out or Compounds Australia website, and also you can contact me if you're interested in this facility. So I wish to acknowledge many people, and highlighted in red are the people involved in Parkinson's research. And Robtima is a new PhD student who is currently working on NMR metabolomics in target identification for Parkinson's disease. Emmanuel looking at establishing the mitochondria membrane potential assays and to test the compounds we have for as mitochondria modulators. And William, Chao, and Chen Ping, and Dong Dong are all graduated from my group. They are embarking on their independent research, and collaborators including Professor Ron Kun, George Malik, and Stefan, who are the biologists helping establish the phenotypic assay. And Jamila helped running the assay and helping the student to establish the assay. And of course, I wish to acknowledge Nature Bank and Compounds Australia for providing the samples for us for this research. And thank you all for your attention.
Thank you very much, Professor Feng. It's really interesting talk. And I would like to summarize your talk a little bit. So Professor Feng, a group working on Parkinson's disease, and right now Parkinson's disease is still lack of understanding. And he used patient-derived cells, which is HON cells, as use it as a window or door to the brain. And he used two types of assay, phenotypic assay and also functional assay, which is for phenotypic assay, she used a different fluorescent tag and using small molecule to study Parkinson's disease. And she used a high throughput screening, which is he screened for many compounds and got hit only few hit compound, and the hit compound isolated from the marine sponge, and she isolated 22 in total compound, which is consists of peptide, alkaloid, and analog of non-compound. And it's really interesting, Professor Feng, thank you very much. It's real wonderful to know about Parkinson's disease, and we are looking forward for the next result from your groups. And right now we are coming up to the discussion sessions, and we have several questions for the speakers. And the first question is from Nuki BRIN. Is there or will be any problem for utilization or commercialization of active compound from marine biota versus ecological matter of the biota? What do you think, Professor Davis and Professor Feng?
Did you want me to go, Yuan, or did you? You can go first if you want.
Go ahead, Rowan.
Could you just repeat that question again? What's the?
Is there any problem for utilizations or commercializations of active compound from marine biota versus ecological matter of the biota?
Okay, I'm not quite sure what they mean by ecological measure of the biota, but in terms of potential for developing marine natural products into products, there's a very high potential, and if you've collected the samples correctly and you've had the scientific permits in place, so you haven't just gone and collected whatever you want from whatever country you want. If you've collected appropriately, ethically, you've got benefit sharing agreements in place that your government can support or is willing to support, I think there's no problem at all. And there's many marine natural products that are out there as commercial products that have made it to the marketplace. Ectinocidin 743 is one. You've got conotoxin, which is used to, or conotoxin derivatives, which are used to treat pain. You've got ARA-A and ARA-C, which are sponge-based nucleosides that are used to treat viral infections as well as anti-cancer. So there's a host of marine natural products that have either themselves or derivatives of the marine natural product have made it as a commercial product. And as long as everything's in place to acknowledge where the genetic resource, the original biota came from, I think there's there's no problem.
Yeah, sure. What do you think, Professor Fine?
I think I tend to agree with Rowan. I think at GRID we have quite a well-established agreement, benefit sharing. I think if anything need to be developed by commercial entities, we will have some agreement in place, and I don't see any major obstacles in terms of developing marine natural products into a drug. There's so many examples out there. Natural product, in fact, is a very, very rich resource for lead discovery and demonstrate it is a quite hot researched area. I don't see any problems.
Sure, but how do you think if we synthesize the drug lead compound from marine natural product? I mean, it is one of the solutions for commercializations. How do you think? Do you agree with it? Synthesis, total synthesis, like Professor Davis did with his compound.
Well, one of the major issues for natural product is the supply issues. So we can isolate molecules with fantastic activity, but if we can't provide enough material for further testing, we can't progress any further. One of the solutions, of course, is the synthesis, synthetic routes to provide sufficient amount of natural products for drug discovery. So I'm all for it. And of course, it's tedious, it needs a lot of effort, and it's not easy to do. But like Rowan has demonstrated, he's very much into making analogs of natural product. And some of his analog have shown quite nice activity. So this is, I guess, very important strategy to overcome the supply issues.
Thank you. And the next question for Dr. Bayu. What an interesting and insightful presentations. Are there any alternatives for comprehensive database of marine biodiversity along with their list of bioactive compound which we can currently use all its feature freely, since main database for marine biodiversity like Marine Lead and DMNP are only accessible by paying subscription fees? Thank you. Actually, I apologize instead of Dr. Bayu. So he is in something urgent matter, so he cannot join this question, but I will answer instead of Dr. Bayu. So right now in our country, we have a problem for accessing a kind of a marine lead to analyze to identify our isolated compound, so it's still a trouble. But actually we have some collaborator that we can ask for help to help us to identify, is this new compound or this is known compound. So we hope that in the near future we can have our own kind of marine lead that can be accessible freely by Indonesian people. And then the next questions, according to the slide, what are the biodiversity workflow at GRID? This is for Professor Davis.
Yeah,
what are
sure.
the biodiscovery workflows?
Yeah, I think I had a slide halfway through my presentation that maybe it jumped to the next slide. It was timed slide, but hopefully the workflow's fairly standard. We take our biota and we've made these libraries, both the extract and the fraction libraries. They're stored at Compounds Australia. And so when someone has a really cool drug target or an agrochemical target that they want to find an inhibitor of or an activator of, for example, and they've developed a nice in vitro high throughput screen, they access these libraries, they screen them, and then a lot of the times they'll do a secondary assay after their first assay run to confirm, for example, that these extracts or fractions aren't super toxic, for example, or they don't rupture cell membranes. You know, hemolysis is not an issue, for example. And then once we know what the active extracts and the active fractions are, the chemists like myself and my team will go back and do large-scale extraction and isolation work. And typically I showed one slide there that had an example of bioassay guided fractionation. And we extracted this sponge. It was a Citronia species. It was towards the end of my talk. We extracted 10 grams. We got about 1 gram of crude material. And then we fractionated using C18 beta cell, which is a reverse phase packing. and we collected 60 fractions, and then we sent those 60 fractions to our collaborators. They tested it and pinpointed where the activity was. And we keep on doing that process until we get pure compounds. And then at the end of the day, you have to work out what the structures are. And I'll just make a comment about, marine lit is very expensive. But the other thing that my students have used when we don't have access to marine lit is SciFinder Scholar is also a great way to do dereplication. And most universities have a subscription to CAS that enables you to do searching on SciFinder Scholar. So we do dereplication of the active molecule to work out if it's a known or a new compound. If it's new, we have to work out how it's new, and we do that by mass spectrometry, UV, crystallography, as well as a lot of NMR. Then we come up with our compounds, and that's when a decision's made. If it's a commercial product or commercial pathway, there's discussions around patent technology, patenting that compound. If it's an academic collaboration and we want to write a grant or a paper, we go down that route. So that's a long story, but that's the biodiscovery process. We do total synthesis, as you saw, where we can, but for complex syntheses, involving 20 or 30 steps, that's not what our group is set up to do. The thioplocortone story was very nice because we could get the job done within 8 to 9 steps. We'll try that sort of synthesis, but not, say, the eribulin mesylate synthesis that everyone knows about is 50 plus steps, I believe, to generate that marine natural product derivative that's based on helicondrin B, which is used as an anti-cancer agent. Hopefully that answers the question, Penny.
Thank you. Professor, I have one question about this matter. So you isolate this active compound, but what if you isolate active compound, but actually this is for example, as anti-malaria, but this compound have a high toxicity. So what is your strategy to modify these structures? Do you use something else? Modify or something? If you find active compound, but toxic one.
Yeah, that was a question for me, was it?
Yes.
Yep. Okay, so if in the anti-malarial world, you really, MMV, Medicines for Malaria Venture, have a hit criteria for their pure compounds. And we were sticking to their criteria. And the initial hit was just a selectivity index of greater than 10. So that can mean that the compound does have some toxicity at some higher doses, but you want it to have a more potent activity against the malaria parasite. It's a little bit of luck involved. I do read about people synthesizing out toxicity from a molecule, but for some natural products, they are inherently toxic, and sometimes the program just needs to be stopped because they have too much toxicity, and it doesn't matter how much med chem you do on it, you're never going to remove that toxicity profile. We've had a couple of successes some of which we're going to publish in the near future, where we've taken something that's got moderate toxicity, added a couple of other functional groups using late stage functionalization. We do a lot of that now. And so this is semi-synthetic work, essentially.
Wonderful.
And we've found compounds that are less toxic and just as active against a parasite or a bacteria. So you need a little bit of luck in research, as you know.
Excellent. Thank you very much. For the next question for Professor Davis again. So thanks for your presentation. My name is Alvi. I'm a lecturer as well as researcher in West Nusa Tenggara, Lombok Island. I am also Australia Award Scholarship awardee for doctorate degree for intake 2023. I conducted some research about anti-malaria as well. I'm really into grit and Griffith Nature Bank since I saw it in Griffith profile. I would like to pursue my PhD in grit by my research proposal that I have conducted about anti-HIV from echinoderm. Is it possible for me to apply my PhD program at grit? May you give me some suggestion? Thank you.
Yeah, sure. Okay. So, yes, we do. It is possible to apply, and it's quite a long process for international students to apply, but Griffith offers international PhD scholarships every year. And now that COVID is, settling down a little bit, it's getting easier to, start these projects in person. So there's a wealth of information on the Griffith Uni HDR website about how to apply, but that this person can go and check out, or they could email me at my Griffith Uni email address, and we can have a discussion. These international scholarships are very competitive. So you've got to have a high class master's degree or an equivalent to an honours degree, and GPAs have to be pretty high. Master's theses have to be marked and have a score. So it's a long process, but yes, it is possible.
Okay. Good luck for Ivan. Okay, so for the next questions, this is from Sena, where was the Haemonococcus contortur parasite found? Thank you very much. I think this is for Professor Davis.
Yeah, Haemonococcus contortus is a parasite that lives inside sheep and goats, small ruminants. So it lives in the guts, in the intestinal lining of these animals. And I showed one slide that I went very quickly through, but it was the life cycle. So what happens is these parasites, they breed, they reproduce inside the organism, the host organism being the sheep, for example. They excrete eggs that are then in the feces from the animal. They're pooped out into the pastures, and then these eggs are then eaten by other cattle when they're feeding on the grass, and then the eggs get ingested, and then the life cycle continues. And there's a couple of different larval stages. It's very complex. But yeah, they need the sheep or the goat or the small ruminant to survive these parasites, but the eggs can last for a long time in the paddocks and pastures, unfortunately. That's the big problem.
Okay, thank you very much, Professor Davis. And next question also for Professor Davis. Thank you thank you for very informative presentations. I am interested to the extract, fractions, and natural product library from marine organism. Quality control of the sample in the library is probably one of the most important issues to ensure the quality of the obtained data. Could you inform us how do you manage the quality of the sample in the library, especially sample in form of extract or fractions?
Yeah. Okay. So, yeah, great question. It's all about, there's no point sending out samples if they've degraded or they're no longer containing the natural products you hope they might contain. With all of our samples, we don't know what the compounds are in these extracts and fractions, but we do have, when we were working with AstraZeneca, they gave us a lot of advice about storing extracts and fractions, particularly in dimethyl sulfoxide. And over a number of years, we did a lot of quality analysis, QC analysis, on a small number of extracts, both marine and plant. We never published this work, but it was doing things like UHPLCMS profiling. And we showed that over a course of many years in DMSO, the majority of samples maintain their integrity. But what was discovered was that after 5 years or more, the stability of some of these compounds started to drop off a little. So what we do at Nature Bank to get around that, so we've got over 135 samples. We cannot do UHPLCMS on every sample every year. So what we did after doing those earlier degradation studies was we decided that we would throw the library out every 6 to 7 years. And then we have to regenerate the library. So that's a very costly thing to do.
Yeah.
But that was the advice from the pharmaceutical company that we were working with at the time. And that's what we try to stick to. Yeah. With our pure compound libraries, I should comment, and this is Compounds Australia, who store the open access libraries that Ewan and I were talking about. They take random samples every year and they do LCMS analysis. And then they give a report to the person that owns those libraries, telling them how the compound's lasting and how stable it is. So that's a lot easier to do when you're dealing with pure compounds as opposed to extracts and fractions. But Compounds Australia don't do LCMS on 1.5 million compounds either. They only do a subset of those libraries. Yeah.
Okay, thank you very much.
Just to supplement on that, I think when we take compounds from academic groups and we do the LCMS analysis first, and then from then on, later years, we just randomly choosing samples for quality control.
Yes. Great, thank you, Professor. And for the next question is from Mrs. Ani. I would like to ask a question to Professor Yun Feng, whether your group also use any other neurotoxin such as 6-hydroxydopamine, 6-OHDA, or MPTP for your analysis to screen potential compound for Parkinson's disease therapy.
We do. That's a very good question. We normally use 2 types of positive control, and rotenone is one because we understand it quite well. We know how it works, but the problem with rotenone is its solubility issues. It's very hard to solubilize to have higher concentration, and therefore we do use a 6-OHDA. That's the 6-hydroxydopamine. We do use chloroquine at times. And we also use MPTP as well. So for the mitochondria membrane potential, we're not going to use rotenone. It's likely we're going to use 6-OHDA.
Okay, thank you very much. And for the next questions, I think this is for both professors. How to access the Australian Griffith Chemical Repository? Is it free access or agreement-based using? This is from Nuki BRIN.
Well, maybe I'll start, Ewan, and I'll just talk about the Nature Bank access because, and then Ewan can talk about the other libraries that are held at Compounds Australia. But for Nature Bank, there is a small access fee that's requested, as well as, of course, a material transfer agreement, and that's. The access fee is based on a pro rata rate. So, if there's a certain access fee for wanting the 105,000 fraction library, and there's a different access fee for wanting only 1,000 fractions, for example. The other option that can be done is that if people are interested in only, say, one particular plant, or a particular marine species, and we have those in our library, we can do a fresh extract of those and ship those. But we are not allowed to legally ship the raw biota, which contains the DNA, of course, that can be used for other purposes. So, yep, it's all possible, but it's normally, it's a negotiation that's on a case-by-case basis. The collaborators have to also pay a plating and shipping fee that Compounds Australia comes up with, because it's very costly to send microtiter plates around the world. It's not a cheap exercise.
Well, for compounds deposited in Compound Australia, we normally have agreement in place. If you want access to compounds, we will send you the standard agreement, and there is a small fee involved in access to compounds, but they really cover some very basic consumables, plating cost, and shipping cost. So it's not hugely expensive, but there is a small fee involved.
Okay, thank you very much. So there is one raised hand. So let us audience to ask directly. So please, to the team, could you? Oh, okay.
Okay. So can you hear my voice?
Yes, sure. Please.
Okay. So for Professor Yun, I'm the one who asking about the 6-hydroxydopamine and PTP. And thank you very much for the answer. But I would like to further ask the similar question actually. So you are using the cell from the human olfactory Parkinson disease, I mean, from the patient. So have you ever wondered, I mean, do you know that probably we also can get a primary cell culture from the enteric gut? I mean, because it's also the enteric nervous system from the gut. It's also, I mean, maybe related to the hypothesis that probably is also the source for the toxin in Parkinson's disease. So thank you.
Well, thank you for that suggestion. We have read quite a few literature in terms of linking neurodegenerative disease with gut health. So there's quite a few reports in that. We have not really started to using different cell lines. So one thing is to access how we get them. And secondly, we are very familiar with Horn cells. We have done quite a bit of research. We know how it behaves, the link between Horn cells and Parkinson's disease. But that does not mean we're not open to use other cell lines. For example, mitochondria potential assay we are currently developing, we want to use a different cell type first, then move on to Horn cells to see whether there is some differences between different cell lines. So we're absolutely open to using different cell type. And thank you very much.
Thank you so much. Okay.
Thank you very much, Mbak Dwi and also Professor Feng. And the next question is from Tresia Sandalayuk. I want to ask about the anti-cancer research method extracted from macroalgae, especially looking for phenolic compounds. What method is suitable to use? Thank you. I mean, maybe this is for both professors. They want to isolate anti-cancer compound, especially phenolic compounds from macroalgae.
Mm-hmm. You want me to go, Yun, or do you want to go?
I can sort of start with very brief. I don't do much anti-cancer research right now, but in terms of phenolic compounds, I guess you're asking how to extract it. That's maybe my understanding. If I understand not correctly, let me know. But in terms of extraction at GRID, Ryan would perhaps say a bit more. We use sequential extractions. We use a non-polar solvent first, then move on to more polar solvent, and firstly to defat the plant or marine organism to extract the very lipophilic compounds. Then we use dichloromethane and methanol to targeting the mid-range polarity molecules. So phenolic, it's a large group of compounds. It can be really polar, can be even water soluble. I think you really need to consider what solvent you use when you think about the class of compounds you want extracted from your plant or marine organisms. So that's my 2 cents worth of advice.
Yeah, that's pretty good advice. I'd just add that, with the samples that we look at that are polyphenolic, anything that Nature Bank looks at, we've got dry biota. So we don't have wet things that we don't go and collect a sponge and then bring the wet sponge back to the lab and try and extract it. We freeze dry that sponge, make a dry powder. We freeze dry it, then we grind it into a fine powder to increase the extractable rate. So we want to get as much of the natural products out of the sponge or the coral or whatever, or the algae. And we do that by making very small amounts of material. And that raw biota is extracted, as Yun said, with hexane, dichloromethane, methanol. And I think so we've got a very standard protocol, and that's it, hexane, DC, and methanol. But like Yun said, if you think that the phenolics that you're working with are highly water soluble, you should also throw in a water extract or a butanol extract. Ethanol also is a very nice extracting solvent. we've had in some of our past polyphenolic projects, we've actually, from the dry material, we've actually used acetone and only acetone to extract. Acetone can be a little reactive sometimes, you've got to be careful, but I think there's a bit of trial and error required like on a case-by-case basis as well. So sometimes you have to alter the solvents that you're using and even the temperatures that you're using sometimes to get the best extraction rate.
Okay, thank you very much. I think this is the last questions and time is up. Thank you very much for Professor Davis and Professor Frank. It's really wonderful talk and very warm talk, and we are very lucky to have you here. And thank you very much. And we hope and we are looking forward for your future result. And we are really, how can I say, we are really interested in your research, and we hope that we can do more collaboration in the future. We don't know that. We hope we can contact each other. And thank you very much for your great talk today. And I will give it back to the our MC right now, Miss Asri. So floor is yours. Thank you very much. I'm Penny.
Okay, thank you for sharing your vast expertise and information with us today, Miss Penny and all the speakers. I believe that it gives us a wealth of materials to improve our lab skill, particularly our research abilities. Before we finish our seminars today, I hope the speakers and the moderator don't mind if we take a picture together. I'd like to ask the committee to assist us in taking a picture.
Sure.
Please. Okay, I will count from 1 to 3, okay? 1, 2, 3. Once again, maybe? Yep. Okay, once again. Okay, 1, 2, 3. Thank you all. Okay, we have officially concluded the webinar and would like to thank everyone who participated and support us during this session. But don't forget to come back again for our second sessions on Monday, September 5th, 2022. We will have more incredible speakers at the time. You will regret if missing it. And as a result, we hope to see you there. Have a fantastic day. Thank you very much. Thank you. Bye.
Thanks, everyone.
Thank you, Prof. Fang.
Thank you very much.
Have a good day.
Also,
have a good day. Take care.
Yeah. Have a good day all.
Bye-bye.
Bye. Makasih ya Bu Penny. Terima kasih juga kepada Bapak-Bapak dan Ibu serta teman-teman sekalian para audiens yang telah hadir mengikuti acara webinar MERIN kita pada hari ini. Sampai bertemu kembali pada kesempatan berikutnya dengan tema yang sama, masih dari kelompok riset MERIN yang akan hadir gitu pada hari Senin pekan depan tanggal 5 September 2022. Saya Asri sekaligus mewakili panitia yang bertugas pada hari ini berterima kasih sekali lagi dan juga memohon maaf kepada semuanya jika ada yang kurang ya dari kami. Baik, akhir kata saya ucapkan wassalamualaikum warahmatullahi wabarakatuh. Sampai ketemu lagi. Salam.